Nearly 40 years ago, I fell in love with neural networks. This is that story. At its core, a neural network is simple. Inputs, weights, a summation, an activation function, and layers of units that learn from examples. Inspired by NETtalk, in which a neural network learned to read aloud, I engaged in several neural network projects. It started in the late 1980s at the Colorado School of Mines, where I split my time between chemistry and computer science. I had a lot more hair back then. One of the first projects taught a network to read mass spectra in separate plant pathogens like Xanthomonas from the ubiquitous Pseudomonas. From there, I moved into protein structure prediction. The first project trained a network to predict whether a cysteine in a protein would form a disulfide bond or stay a free thiol. We fed it the amino acids flanking that cysteine and let the weighted network learn the pattern. Next, we asked whether we could predict if any amino acid would be buried or exposed on the protein surface, straight from sequence alone. Certain residues, like tryptophan, carried a strong signal for burial that showed up clearly in the trained weights. That work grew into predicting a protein's secondary structure content with a tandem neural network, what we now call an early form of deep learning. One network estimated helix and strand content, and a second network generalized across proteins to sharpen that first guess. It was a genuinely overdetermined problem. Fewer than 200 solved protein structures existed at the time, and our networks had far more adjustable weights than we had training examples. We had to invent our own tricks just to prove the network was learning real structure and not simply memorizing the training set. All of it led to my final PhD thesis at Berkeley, predicting features of protein structure with computer-simulated neural networks. In its opening pages, I wrote that those who provide a means of predicting protein structure would undoubtedly share a Nobel Prize. That's exactly what happened 30 years later. Remember those 200 protein structures I had to work with? They had 200,000 and a whole slew of additional sequence information across species. But that is another story. From Berkeley, I moved to MDL, Molecular Design Limited, where my work helped push out the release of their MAX key set representation. I built a system called ASSALT, Automated Substructure Activity Learning Technique, using neural networks on those MAX keys to predict bioactivity across the drug data report and the comprehensive medicinal chemistry databases. It became a showcase application for IBM's new RISC chip, part of the IBM and MDL partnership that put ISIS/host on that blazingly fast new platform. Here's me in the MDL era with noticeably less hair. After MDL, I joined Affymax, where we used neural networks to model chemical structure against biology and dug deep into ADME, especially human intestinal absorption, inside the Affymax glaxocombinatorial lead optimization program. There we coupled a genetic algorithm with neural networks and a reinforcement learning strategy to pick the best descriptor set for predicting absorption. Foundational work we later reused for other optimization problems. The training and cross-validation sets lined up well against the observed absorption values, and the external prediction set held up too, validating the model beyond its own training data. Ironically, the hard part was never the algorithm. It was the many, many months spent curating the literature data itself, which is part of why this remains one of the more highly cited papers in the field. Then, at Sertanty, later Eidogen-Sertanty, we explored synthetically accessible, medicinally relevant chemical space, again pairing a genetic algorithm and reinforcement learning with neural networks. Those networks predicted potency and cellular activity across entire kinase panels. We mapped out which chemical transformations and building blocks could realistically reach a target, not just which ones looked good on paper. It let us ask what can you make and what should you make in the same breath. For nearly a quarter century since, we've kept collaborating with biopharma partners on neural network models in one form or another. Here's me in 2012, even less hair by then. Then, large language models exploded onto the scene, and natural language coding tools changed everything. We used to write a few million lines of engineered code per decade on projects like these. Last year alone, we shipped 10 million lines or more. That leverage opened up new ground. Apps that help monetize content, celebrate friends and family, add bionic productivity to my daily work, and take on boil the ocean projects in the food is medicine space. It also fed a few content hobbies of mine, mixing music videos and posting them out across platforms. That's me now behind the drum kit more often than not. I'm Dr. Stephen Muskall. Come join me in the Renaissance Circle.